Welcome to our virtual space, where thought leaders who, in a variety of ways, have committed themselves to improving our lives share their work, perspectives on current affairs, and what brought them to where they are today. My name is Rob Liu, and this is The Xchange. So, Bavesh, thank you so much for joining us on The Xchange. You have done an enormous amount of work in South Africa on tuberculosis, and so your insights into infectious disease, and in particular infectious disease on the African continent, I believe are quite profound. And so one thing I know, though, that our listeners and viewers would love to get a clearer sense of is, in terms of all the fantastic work that you've done in terms of your life and career as a research scientist, what actually brought you to that point? What brought you to that career? So if you could share with us a little bit what was the pathway that led Bavesh to who Bavesh is today?
Thank you very much, Rob. It's indeed a pleasure to be here and to be able to speak to you about what we do here in South Africa. I'm a TB researcher. I became interested in tuberculosis as a child. So as a child, I grew up in South Africa during the time of segregation. From a political perspective, South Africa was experiencing apartheid. And my dad was a farmer. Being of Indian descent, we were not allowed to own any land. And so what my dad would do would be to rotate off land from different farmers. And during a particular rotation, we would grow a crop and then harvest the crop and move off to another place once the crop was harvested. And we had a very small group of farm workers who used to move with us around the country as my dad did his farming. I mean, they were almost like family. But slowly and consistently, somebody would disappear in a season and then the next person would disappear. And I would ask my mom, what's going on? Where are these people going? And she would say, well, a lot of people have tuberculosis and, some of our farm worker friends are dying of tuberculosis. And, as a child, you view disease in a very different way. And, this tuberculosis was almost this sort of unnamed monster that was taking away some of the people in my life who I best identified with as I was growing up. And so as I went through school and university, tuberculosis was something that I always kept at the fore. When I finished school, I wanted to become a doctor. I applied to go to medical school to get into the medical degree, and I didn't get in. But what I did get into was for a degree in science. I saw that as an opportunity to pursue this lifelong passion of trying to do something for tuberculosis. So I ended up doing a PhD in mycobacteriology, where we studied how the bacteria that cause TB, how they breathe, how they use oxygen. And it's still a disease that has fascinated me for a very long time. Based on death certificates, more South Africans die of tuberculosis than any other bacterial infectious disease. Despite the fact that we have combination chemotherapy, we have up to 20 different antibiotics that we can choose from when we treat TB patients. We still have 1.8 million people roughly who die of tuberculosis every year. We have a vaccine, the BCG vaccine, and it's probably the most widely administered vaccine in human history. Yet you have anywhere between 8 to 10 million new infections every year. For some reason, even though humanity has battled with this disease for hundreds of years, we can date back TB to thousands of years in Peruvian mummies and in prehistoric sites. We still haven't been able to eradicate the disease. And it's been something that's, as an academic, as a scientist, as a person, it's been something that just bugs me. I'm sure we can do better when it comes to TB. And so, I did my PhD in TB research. And then I went off and I continued my passion for TB by doing postdoctoral studies in several aspects of TB research. I worked for a long time. I worked for half the year in the US and the other half in South Africa. I was in different laboratories in the US and in a laboratory here in Johannesburg in South Africa. And then eventually in 2010, I started my own lab at the University of the Witwatersrand. And it was focused again on TB research. And basically, I wanted to, when I thought about what would my lab do, I really went back to those ideas I had as a kid growing up. Why is this disease so hard to eliminate? Why is it that we give people medicine and they don't get better? What's the problem? And so I said, well, if you look at other bacterial infectious diseases, what has been so successful in terms of clinical interventions, therapeutic interventions, antibiotics? And the first thing that comes to mind is like something like penicillin, right? Lots of people have taken penicillin, and the introduction of penicillin, into the clinical setting has really changed how we deal with bacterial infectious diseases. And so for those who don't know what penicillin is and how it works, it's an antibiotic, and it works on by targeting a particular component of the bacterial cell wall. And if we think of bacteria, they're covered by these incredibly complex layers of material that protect the vulnerable inside compartment. And penicillin basically helps to break down one of those layers and effectively causes the bacteria to explode. But for a variety of reasons, we don't use penicillin when we treat patients who have TB. Because TB bacteria seem to be inherently resistant to penicillin. And so one of the things I said my lab would focus on is, can we develop a new penicillin-type antibiotic for TB? Something that targets the same layer in the TB bacterial cell wall that penicillin targets in other bacteria. And so that layer is called the peptidoglycan layer. And it's made up of a whole lot of cross-linked sugar-type polymers that create a mesh around the cell, and that gives the bacteria rigidity. And so a fair amount of the work in my lab today is focused on trying to understand how do bacteria remodel this peptidoglycan layer. Because if you imagine you're a bacterium and you need to grow, as you expand, you're cutting this penicillin-sensitive layer to allow for the insertion of new cell wall material. And at that point, when you are cutting and remodeling as a bacterium, when the bacterium is cutting and remodeling the cell surface, it's incredibly vulnerable. So those enzymes and those molecular machineries that are involved in remodeling of the cell surface become very attractive from a drug target perspective. And so one component of my lab focuses on discovering new TB drug targets, but particularly those that would attack the cell wall and behave in a way like penicillin does, cause the bacteria to break down and perhaps even explode. And that work is ongoing. We have several different interesting techniques that we apply to that. We do gene editing of the TB bacteria, changing particular components of enzymes that remodel the cell surface. And then we take the organisms that result from there, and we do lots of different experiments with them, including time-lapse microscopy. At a real time, we see how the bacteria extend and how they divide, and how those processes are perturbed when we've interfered with some of these enzymes that remodel the cell surface.
You have just shared, I think, one of the critical aspects of understanding, if you will, a pathogen, so that therapy can really then flow from that. So in a way, what you've really expressed, I think, beautifully, is how understanding the basic biology of a pathogen, of multiple processes in the life cycle of a pathogen, can in fact provide all manner of windows into ways in which you can try to destroy the pathogen. And so I think you've really expressed that connection between basic science and translational medicine really beautifully.
Yeah, I mean, that's, I think also you hit the nail on the head that, when we look at TB as a disease, as I said, it's been around for centuries. It represents a public health failure of massive proportions, right? And why is it we have that? One of the reasons I would argue is that we have failed to understand the fundamental biology of this pathogen, of this bacterium. And a better, more enhanced understanding will allow us to develop more precise therapeutic interventions that are perhaps more long-lasting. And in fact, you can see, when we fail to understand pathogenesis and basic biology, you can see the very dramatic effects on public health today when you look at the coronavirus endemic, right? We have so many unanswered questions that are holding back public health interventions. When do antibodies against the coronavirus develop? When somebody has antibodies, are they protected? If somebody has viral RNA in their throat, does it mean they're infectious? Sort of these basic things that sometimes fall by the wayside in our drive to, just develop healthcare products, then come back to haunt us. The failure to understand the basic biology comes back to haunt us. And so we've really tried to take this approach in the lab that, the more knowledge we develop and the more holistic our understanding is, the better we can place new health interventions.
Yeah, so it seems as if we look at the the COVID-19 pandemic that we're all experiencing right now and suffering through right now. What's quite clear is that what you've just shared with us about how with TB there's this complex matrix of not just the pathogen, not just the pathology of the actual disease itself, but there's also how research is funded, how public health is really designed and implemented based on our understanding. And how do we think about at a societal level the question of why is it that there's so many therapies, but there's still 1.5 to 1.8 million deaths per year. With that context and that deep background that you have, looking at what's happening now with the novel coronavirus and COVID-19, you've touched on already things we should maybe, pitfalls we should avoid. Are there some other perspectives or aspects that you think we would do well to learn from TB to apply to the huge effort that we're doing right now to try to address COVID-19?
I think a significant challenge the world has had when it comes to the coronavirus has really been around its diagnosis. The readiness level to do diagnosis for a new pathogen like the SARS coronavirus 2 was really not at the level where we would have liked it to be. And in fact, most of the world has been rapidly playing catch up in terms of trying to develop the testing capacity, implement high throughput testing. But remember, testing for the sake of testing is really not useful. It's only useful when you couple the result to very important and precise downstream public health interventions, such as isolation and very careful contact tracing. And I think that, that was something it was a lesson that we learned in TB some time ago, and we learned it hard. Because we know that a lot of TB, especially in South Africa and in other endemic countries, a lot of TB that we see is driven by transmission. And by far and large, you'd be surprised to hear that we know very little about TB transmission. All we know is you have to share air with somebody, and through the sharing of respiratory aerosol droplets, you inhale these TB bacteria, and you eventually develop disease. But when we go down to the nuts and bolts of it, and we ask, well, who's transmitting TB to who? So if we look at a place like South Africa and we say, okay, 100% of our TB cases, can we track down every single one of these? Where did the infection occur? And, the best we can say is roughly 20 to 30% of infections occur in the household. And for the rest, we have no idea how they occur. And the TB field has really been pushing long and hard to try to develop new ways of understanding how transmission occurs so that you can develop novel transmission blocking strategies, right? So interruption of transmission, will have a very dramatic effect in terms of containing a disease and allowing the other public health interventions to reap maximum benefit. And I think all those lessons are really important today when we think about another, infection that you can get by sharing of air, which is the coronavirus. The idea of we need to understand who's infecting who's the most infectious, where are the infection hotspots, where do you deploy the best public health interventions? And in a situation where even the best resource countries have health systems that are experiencing substantive strain, how do you deploy these very valuable and nowadays scarce health resources to the areas where they needed the most? And I think that we can learn a lot from what's been done in TB. We can also learn a lot from what's happened in the TB field about what not to do. But I do believe that those lessons are being learned as the world experiences the coronavirus pandemic as an entire entity. No one has the perfect solution. You have countries that go through different stages of lockdowns. The science around all of this is not clear. It's not perfect. But I do feel like a lot of the expertise that we've had in terms of respiratory and other viral diseases have been brought to bear on the coronavirus pandemic with as lethal precision as is possible.
Mm-hmm. And also, you've started to do some work directly related to COVID-19. Am I correct, Bavesh?
Yeah. So, in addition to working in the lab looking at the cell wall of TB bacteria, my lab has two other big focus areas, and they somewhat relate to how we ended up, trying to assist with the coronavirus problem today. So the first is the idea that I mentioned when we started chatting is, why is it that you have to treat TB patients for so long? Say if you get a bacterial disease today and you go on to antibiotic treatment, you take antibiotics for roughly 5 days, right? And you'll remember if you've ever been on antibiotics, what does your doctor tell you?
The full course.
Right? Finish the full course. And really the hypothesis there is that there are bacteria, if you're infected with bacteria, there are bacteria that will very quickly respond to the antibiotic and they'll die. And then there's a small proportion of bacteria that are in your system that resist the antibiotic. They're genetically susceptible to the antibiotic. They will die, but they just take longer to die. And a small proportion of them hang around for a very long time. We call these persisters. And so I started off wanting to ask, why is it that we need to treat TB patients for 6 months? What goes on in 6 months that can't be achieved in 5 days with TB patients? And so we set out to look in South African TB patients. Here in Johannesburg, we have a lot of TB, and we have a lot of HIV-associated TB also. So we went to our communities and we said to our participants, can you give us sputum specimens when you're diagnosed with TB? And then when you go on to TB treatment for the 6-month time frame, can you actually give us sputum specimens as you're taking treatment so we can take a look to see what is happening with the bacteria in your body? Why are they taking so long to be eliminated? And what we did was, we developed ways of differentiating bacteria that are very easy to grow, they're replicating, they're responsive to the antibiotics, and then those bacteria that are somewhat more difficult to grow. We call them differentially culturable, because they don't grow under certain conditions in the lab and you need to give them some juice and stimulants so that they can start growing. So their culturability is different. So we use the term differentially culturable. And because they're non-replicating, differentially culturable, in a somewhat lazy state, they're not as susceptible to antibiotics as the ones that are replicating. The ones that are replicating are quite susceptible because many of the antibiotics target processes that actively replicating bacteria use. So if you're a lazy bacterium and you're differentially culturable and you're not wanting to grow, you have some sort of inherent tolerance to the antibiotics. And so this was our hypothesis. We went into these clinics in these communities and we took TB patients and we got specimens from them. And what we found was incredibly surprising. A TB infected individual, before they even start treatment, they have large numbers of these differentially culturable bacteria in their sputum. And when we follow those over time, you see that the bacteria that are actively replicating and that you can grow very easily, they actually get eliminated quite quickly. But it's these ones that are in this sort of differentially culturable state or a non-replicating state, they're the ones that hang around for a very long time. And they're the ones that necessitate the long course of treatment that patients have to take. Now, this is important because when we want to shorten the duration of therapy, we need to know which populations we should be looking at in terms of bacterial populations. And so now we've been able to show that if you want to shorten therapy with new drugs, we need to make sure those new drugs actually target these differentially culturable bacteria. And then you can talk meaningfully about treatment shortening. But we then thought, well, if these organisms are not growing so nicely and they're struggling to grow, part of the way we diagnose TB is we actually grow bacteria. We do TB culture. At least this has been the way that it's been done for a very long time. But there's a more newer way of doing TB diagnosis, and that's using molecular diagnostics, which detect the DNA of TB bacteria. But let's go back to using diagnosis and culture for diagnosis. So if these bacteria are not growing properly, there is a possibility that you could misdiagnose people. You'd get a negative result, and that person actually has tuberculosis. But because there are so few organisms and they're not growing well, you're not getting the results you're looking for. So we thought, well, we need to maybe engage our minds a little more around diagnosis. And, we spent some time developing ways of actually modifying, the culture media we use to diagnose TB to pick up these differentially culturable organisms. But as we started working more and more in diagnosis, we got involved in molecular diagnostics. And that's sort of the third component of what we do in the lab. And so for the longest time, Robert, when you think about TB diagnosis, you'll be shocked to hear for the longest time how horrible it was, right? We used a diagnostic test that's over 100 years old, smear microscopy. We take a sputum specimen and we smear it onto a slide, and then we look down the microscope to see if we can see bacteria. Over 100 years ago, when Robert Koch first described the TB bacteria as the causative agent of TB, he used the same method. And 100 years later, we were using the same method. Half the time it's wrong. If you have very few organisms, it misdiagnoses you as being negative. So that's the one way we had of diagnosing TB. The other way was culture. Culture takes up to 42 days to give you a negative result. This was horrible. We had these diagnoses that could be wrong and that made patients wait a long time to be able to get a definitive answer as to whether they're infected or not. So to address this, a lot of work has been done on molecular diagnostics, particularly on detecting TB DNA. And roughly in 2010, the South African government, through its National Department of Health, decided to implement molecular diagnostics for TB. We were using two molecular diagnostics at the time, the Hain line probe assay, and this diagnostic called the Gene Expert, and it's marketed by a company called Cepheid. So the Gene Expert is really a very neat device in that it gives you the ability to do diagnosis sort of in a near patient setting. So you could put it at microscopy level centers and get patients coming into the community clinics or into primary healthcare clinics and giving a specimen and being able to get a result in a relatively short period of time. But so the South African government saw this and they thought this was a great opportunity to deliver more quality healthcare to people. And they decided to invest in the expert, the Gene Expert, as a device for TB management through the National Priority Program. But then they ran into a stumbling block. They bought the first few Gene Expert devices, and then they were hit with the conundrum of how do you verify these instruments, right? When you have an instrument in a central lab, you have a lot of quality control, you've got a lot of training, you've got a lot of infrastructure on that instrument, and it works. But now when you send it out to clinics, and you need to imagine South Africa, you need to imagine some of our settings, and the associated infrastructure and resource constraints. How do we know that when we place a device somewhere, let's say in an informal settlement or near a clinic in an informal settlement, that it's actually performing properly? So let's step back and think about that for a minute. Right? How do we know? If we have a diagnostic device, how do we know it's working? So let's say we put 100 bacteria in there, and if the diagnostic device tells us there are 98 bacteria there, we say, okay, this works. The margin of error is 2%, and I'm willing to take 2% in my national system, and you run with it. You put the device out in the healthcare system. The problem with TB is you can't do that. TB bacteria are dangerous. So we can't send TB bacteria to that clinic in the informal settlement and say, here, use these to see if your device is fit for purpose. We're going to make people sick. And so the government approached us. They said, can you, the National Department of Health, through the National Priority Program, and my colleague Leslie Scott and Wendy Stevens at the University of the Witwatersrand, where we work, they approached us and they said, look, can you assist with this? And so I sat with my team and, we thought, well, the Gene Expert, or in fact any molecular device, doesn't care whether the bacteria are alive or dead, right? It just detects a piece of DNA. As long as the DNA is there, you're going to get a result if it works. So what we did was we developed a pipeline to grow large amounts of TB bacteria, and then we inactivate the bacteria. And we have a way of staining them, and then we use a process called flow cytometry, which allows you to measure single cells. And so we do the flow cytometry, and we quantitate the amount of single dead TB cells we have. We do a whole lot of QC to make sure the cells are dead. And then we take a known number of dead bacteria and we spot it onto a little dried culture spot on a piece of paper. And that little dried culture spot has a barcode. We then put that into a Ziploc bag. It doesn't need any cold chain. It doesn't need any special handling. And we send it to that clinic in the informal settlement. The operator at the clinic receives it. He or she pops out the little dried culture spot. We put a bit of color in there so you can see where the spot is. They mix it with a bit of buffer, the bacteria uncoat, and then that operator will add the bacteria to the Gene Expert cartridge. And then as the Gene Expert is running, we can see the performance of that spot using cloud-based software. And immediately we can say, okay, that device works. That clinic near the informal settlement is now validated. It can take specimens from the informal settlement and it can report the results to the patients. Everything is working properly. And so we initially started off this validation for the National Department of Health. And in 2011, we did it for all the instruments they bought initially. But by the end of 2012, we had done it for the entire country. And then as there was greater awareness for the approach that we were using, the CDC came on board and they endorsed the dried culture spots. Subsequent to that, the WHO endorsed the dried culture spots. And before you knew it, we were sending these to over 20 countries who were using the Gene Expert device in their national priority programs. And so suddenly, from being this little lab that did some really cool stuff on the TB bacterial cell wall, we became a lab that was enabling many countries to actually validate their TB diagnosis. And that was great. But, there were a couple of problems. In the health system, there's always a crisis. There's always an emergency. And usually, somebody would come to us and say, Bavesh, we need new spots, or we need spots for drug-resistant bacteria. Can you help us? And TB bacteria grow very slowly. And I said, yeah, I can help you. But my joke was, I can help you fast but slow. So we can start a culture for you, but it's going to take us a month to grow it. Then we have to kill it, and we have to wait 2 months to make sure it's dead. And then we'll do the flow cytometry. We will measure the organisms. We'll create the spots for you. So I recognize you having an emergency. I can give it to you in 4 months. Which was somewhat, as you can imagine, it was somewhat undesirable. So at some point I said, this is not working anymore. So we sat down again with the team, and I said, how do we make this even faster, right? And so we just extended our thinking, right? These molecular devices don't care whether the bacteria is alive or dead. And so then I asked my team, does it even care whether the bacteria are TB or not? All it needs is a little piece of DNA that calls the test TB. We just need to make sure that little piece of DNA is there, right? And so what we did was we embarked on this approach called biomimicry. And so biomimicry is really trying to mimic what happens in nature, right? And when you look at nature, nature has spent hundreds of thousands of years of evolution solving problems that we face today, right? And so we took this biomimicry approach where we found safe bacteria, that are not harmful, that are stable at room temperature, they're not difficult to grow. And we engineered their genomes just to carry the bits of DNA that get recognized by these TB molecular diagnostics. And lo and behold, when you put those safe bacteria into the device, it calls it TB. It doesn't know the difference. Right. And so suddenly we went from taking 2 months to grow up something for somebody to 2 days. These bacteria are fast growing. It was quick. There were no safety concerns. And so now the biomimicry approach we were able to use to provide products for TB to people who need to validate TB diagnostics. But here's where the COVID twist comes, right? So in addition to doing drug-sensitive TB, I'm sure you're aware that the world also faces a huge problem of antimicrobial resistance, this so-called AMR problem. A large proportion of that AMR problem is due to TB. There's a lot of drug-resistant TB out there, and many countries who are battling with this, they also need verification material for drug-resistant TB. And it becomes somewhat challenging for us when someone says to me, Bavesh, I want a validation strain for extremely drug-resistant TB that's resistant to many antibiotics. It becomes a bit challenging for us to grow something that dangerous in the laboratory and inactivate it. Yeah. And so we said, well, can we actually repurpose our biomimicry approach to mimic drug-resistant TB? And we just put in that little piece of DNA where you have the drug resistance mutation. We just swap that into the genome of a safe, non-infectious bacterium, and now it reads like drug-resistant TB. And then none of the safety concerns that are associated with that. It's non-infectious, it's safe, it's easy to use. And so we've started to do this for drug-resistant TB. But when the coronavirus pandemic came to South Africa, one of the big things that South Africa, and I think many countries around the world have faced is, how do we get appropriate controls to make sure that the diagnosis we're doing and the diagnostic tests we're running are actually correct? Coupled with that, it's important to recognize that one of the things that the coronavirus pandemic has done, it's also created this big surge of new tests entering the market. Many companies have repurposed their production pipelines, have repurposed their IP to produce coronavirus tests. And, as a health system, when you receive these new tests, how do you know they fit for purpose? How do you test them? How do you know they work? How do you deploy them in the health system? Now, a coronavirus test works if you put coronavirus inside and it tells you, hey, I find coronavirus. But again, we come back to the same problem. The coronavirus is very infectious. It's airborne. How do you work with this? And it's also hard to get your hands on the coronavirus. So we went back to the drawing board and we said, this biomimicry approach that we use for TB, can we actually use it for the coronavirus? To develop an encapsulated control. So it won't be the coronavirus. It will be a bacterium. It will be a bacterial cell wall. But inside of it, it carries genetic material that looks like the coronavirus. And as new kits and testing reagents become available, we can provide this control to people to say, if you're looking for a positive control to see if a kit is fit for purpose, we can give you this. It will allow you to assess whether the the extraction process, where you've got to break the coronavirus surface and get the genetic material out. That genetic material is then used for testing using polymerase chain reaction or PCR. So you have two separate steps. It will allow you to actually determine whether your new test is fit for purpose for those two particular steps of the testing strategy. And so, we started this process in March, and we used a target that was available from the CDC and the WHO for a large number of tests that were already out there. And we engineered our bacterial biomimetics to start to look like coronavirus. We provided these to our national testing system, and that enabled them to start to validate tests and to actually start rolling out testing, on a mass scale. And it's really very simple. It becomes like all we do is we grow up these bacteria. They take about a day and a half to grow. They carry these coronavirus targets. We inactivate them just to be absolutely sure, even though there's no safety concern, because the inactivation also stabilizes them. And we just send them out wherever testing is being done. And we say, if you want to validate your test, if you want to make sure your workflow operates correctly, if you want to put a little small car or a truck outside of a mine, and you want to do testing in a remote level, and you need some controls, here are controls. So it's an enabling reagent that allows people to develop, quality assurance and proficiency testing.
Yes.
And so we started our coronavirus work using this biomimicry approach. And then we sort of got into the sort of more hardcore stuff. So, you'd be aware that coronavirus testing can be done in two basic ways. You either detect the virus or you detect antibodies to the virus in people. So we call the antibody detection serology testing or antibody testing. And there are lots of antibody testing kits that are coming out onto the market because these are more easier to use in the home. They're more easier to use in the remote settings. You don't need any lab infrastructure. You just put a drop of blood and you can get an answer. But for validation of antibody tests now, we can't cheat antibodies. Antibodies are very specific. They'll know that we have a bacterium and not a coronavirus. And so for that, we really need the coronavirus. And so a few weeks ago, we have these biosafety level 3 containment labs where we do our TB work. We repurposed this laboratory to actually start to grow coronavirus. And so we grow coronavirus on mammalian cells. You develop a monolayer of cells on the surface of a petri dish. And then you add an aliquot of the coronavirus, and then it infects those cells and multiplies in the cells and then gets secreted out into the medium. And so we set this up. It took a bit of doing to repurpose the personal protective equipment and all of our safety protocols. Yes. And last week, we were able to harvest our first batch of viable coronavirus. And we're now providing this also to the national testing system to use as an additional control, either for PCR-based testing or for serology testing.
Yeah. So I think this is enormously exciting, Bavesh, and kudos to you and your research group for doing this. I think really you've raised a point that is not really discussed at all, which is the importance of validation, right? The importance of positive controls and the complexities that exist around that with a pathogen that is easily spread and is quite difficult to get a hold of in some ways. And at the same time, I think you've also shown sort of the power of really biomimicry, creating a much safer alternative for creating that kind of a validation protocol. So kudos, absolutely brilliant. I think it's fantastic. So Bavesh, one thing I wanted to do is, I think for all of us, there often when you think about our pathways in our lives, there are forks in the road. And sometimes you go left and sometimes you go right. And based on the choice that you make, you end up where you are today. So I'm curious if in all the different crossroads that you pass through, from your childhood to now, was there a crossroad where if you had gone left instead of right or right instead of left, basically the Bavesh would be talking to today would be doing something completely different? And if that's the case, can you share with us in a parallel universe what that Bavesh would be doing?
That's a very interesting question. As I said, growing up, I wanted to help people who had TB. And I must admit, as a kid, and even as a kid in high school, you have quite simplistic views of the world, right? And the only way I saw myself doing that was being a doctor. And not getting into medical school, that was a point in my life where I had a fork. There were other things I could go off and do. I come from a family that has a strong interest in farming and commerce. I could go off and do that. Or I could go off and do this career in science. I chose the career in science, and it's been a wonderful, wonderful journey. Science, fundamental research, exploring biology, learning how the world works has really been, for me, very satisfying. And it's given me the opportunity to take these basic ideas, turn them into really cool research, and then take that research and translate the whole body of work that I've done over a long time into actual useful things that people can use every day and allow them to access better healthcare. But I must admit, even as I went through my PhD, when I finished my PhD, I was faced with, do I do a postdoc or do I go off and perhaps work in a company, develop a sort of more commercial career? And I must admit, the allure of that was very enticing. I had a couple of options to pursue what you would call a career postdoc.
Right.
And actually develop a career in industry, and maybe I would be working in a large pharmaceutical company now. But again, I have no regrets staying in academia. I've also had the privilege to work with wonderful mentors. I did my PhD with Valerie Mizrahi here at the University of the Witwatersrand in Johannesburg. And she really inspired in me this love of science. And when I was in the US, I worked with Dr. Gila Kaplan, who was at Rockefeller, and then went off to the Public Health Research Institute, and in fact became the global director of TB at the Gates Foundation. And she too inspired me in many ways, to try to make a difference. Whenever I went to her with a problem, she would ask me, well, if you had the answer, what would it mean? She always forced my thinking. And in fact, I spent some time at Harvard too, with Eric Rubin. And Eric Rubin, Eric Rubin he's just one of those guys that has such a love for science that it's infectious. And when you're with Eric, you can't imagine doing anything else except science. That's the beauty of a guy like that, And so, I'm glad that I chose the road that I did at those different forks, because they brought these wonderful people into my life. And then I'd be lost without my team. I've given you a 30-40 narrative of the kind of science that I do, but I really want to emphasize that, I'm one person. I sort of sit in my office these days and do very little lab work. I have a wonderful team of, graduate students and postdocs and researchers who've come into my life and really enriched me in many wonderful ways. But, the thing that they've done is they've bought into my thinking around TB research, and they've really driven it and taken it to a different level. So, I really want to acknowledge my team. It's a group of wonderful people. And, you go to work, you do this great job, and you have these wonderful people that you work with, and who could ask for more? So, I hope that answers your question. It's almost hard for me to imagine myself doing something else when you have so many wonderful things going for you in the path that you've chosen.
No, of course. And also, it's fair to say that even though you mentioned that you might have gone into sort of more of a commercial sort of direction, from the work that you're doing, I'm sure, and it's quite clear, that you do have interactions with pharma, sort of, etc. So in a way, it's not as if you're cut off from that world. You just have a different relationship with that world. So in some ways, that element, I'm sure, is certainly still there. But so
you could say I'm living the best of the best of both worlds. But also, the thing that, and I think science is changing, and the way we view science has to change too, right? When I was a postdoc and when I was a graduate student, it was good enough to do science for the love of science, right? And today, just again, look at the coronavirus pandemic, right? The love of science is not enough. When you engage and grapple with a problem, it's got to be solutions driven. And I think that having a more entrepreneurial view of science, it encourages thinking that has greater latitude. It's more relevant. And as youngsters come into science, graduate students and postdocs, they come to me today and they say, well, I want to do something that has meaning. I want to do something that has impact. And so in a way, having this sort of, one arm in a commercial space and one arm in a basic space, I think it allows me to attract a lot of good students, but also to give a more balanced outlook to the scientific world, to the people who train with me. So thank you for pointing that out. It is like having the best of both worlds.
Yes, absolutely. So, you've shared with us earlier sort of your very interesting path. And Here at LabXchange, one of our taglines is learning without limits. And arguably, we've all experienced hurdles, barriers, or limitations that we had to overcome to basically end up where we are today. So I'm wondering, would you mind sharing with our viewers and listeners, was there one hurdle in your pathway that you had to overcome prior to you being where you are today?
One hurdle. Oh dear. There were many hurdles, but there are a couple that stick out. When I went to school, at the time, as I'd mentioned earlier, South Africa was still segregated. And so schooling was separate depending on racial categories. So the school I went to was in an informal settlement. It was a wonderful school. I had really wonderful, committed, brilliant teachers who did the best that they could with what they had. But I must admit, when I got to university, first of all, I was dealing with the disappointment that I didn't get into medicine, and I was doing a science degree. I felt very underprepared. I would sit in lectures, I would sit in courses, and sometimes what the lecturer would be saying sounded like Greek. Like I was sitting in a math class and I thought, this can't be math. I've never seen this before. And I turned to the person sitting next to me and I said, do you know what's going on? And they said, well, we did it in our final year of school. And I thought, but I didn't do this in my final year of school. As I went through that first year, your first year at college or universities is very defining in a number of ways, because you break the boundaries of school, you're more independent, and kind of it's really up to you whether you make it or break it. And so I went through this first year, and as the year progressed, I just felt heavier and heavier. I was failing in class. As much as I tried, I was either failing or just barely managing to pass. I used to do reasonably well in school. I wasn't accustomed to failure. It was something that I was not mentally ready to deal with. And as I failed again and again and again, rather than developing the skills to deal with that, I just sort of curled up inward saying, oh my God, this is turning into a huge mess. And at the end of my first year, I barely managed to pass. I'd failed some courses. And, life looked very, very bleak. And I just thought, well, maybe I should give up this university career. I'm not made for it. Affording university education was also a problem. It wasn't like my family had infinite resources to continue to send me to university. They were really reliant on me getting bursaries. And my academic record did not look like one that would attract a lot of bursary funding. So, it looked like I was done. And I remember that at the end of that academic year, when you go and you look at your marks and they're up on this notice board, and I looked at this and I felt like my chest was going to explode, because these marks were not me, but yet they had my name next to them, and I didn't know what to do. I walked away from varsity thinking that was it, I'm done. But then in the new year, the university invited me back to continue with my degree. They said, look, you failed a couple of courses. Let's see if you can't do some concurrent courses. Let's see if you can't get some assistance and get going again, And my journey was like this. I remember when I went back to varsity, I went to this, I hadn't done well in maths. And I went to this notice board in the maths department, and I was looking for anyone who was offering tutoring, and I still wore this horribly dejected look. And a lady walked past and she turned to me and she said, it's only the second day and you look horrible. What happened? And I said, no, I tried really hard. I worked and I still got these lousy marks. And she sat there and she let me rant. And then she said to me, you can do one of two things. You can curl up and, cry, or you can get over this. You can channel all this rage that you're feeling and you can get over it and try to actually improve your marks. Like, okay, so, life wasn't fair. You didn't get a fair shake, right? But you have to fix it. You can't wait for somebody else to fix it. And she was amazing. She was a PhD student in the maths department. And she said, I'll meet you on the steps of this building, every Monday, Wednesday, and Friday during lunchtime, and I'll tutor you. No fee, no nothing. We'll find an empty classroom and I'll tutor you and I'll help you. And she did that. And I went from barely passing maths in that year to getting a distinction at the end of the year. And the same thing happened for me in chemistry. I was completely lost with chemistry. And one of the chemistry demonstrators in the laboratory sessions came to me and said, I can see when people are confused and you're way beyond confused. What's the story? And I said, I'm missing so much fundamental knowledge. And she said to me, every morning I get in here early on the bus. I see you on the bus, which is why I've come to speak to you. Every morning I'll meet you outside the bus, and as I have my breakfast, we'll just go through some basic things, and let's see how we can help you. And again, I went from barely passing to getting the highest mark in the class. So, I did get to that point where I thought, oh my God, I'm done. Like, I can't get past this. But, I like to say I'm the product of many people's kindness. It's what you see in front of you today is the product of a lot of people's kindness and very generous input into helping me when I stumbled. And so that's kind of how I got over some of the hurdles that I faced.
Oh, what a fantastic and really touching story to share, Bavesh, because I think it really underscores that, as you said, that kindness, that willingness to help others can have a dramatic impact, not just on that individual, but look at the work that you're doing, right? And really the role that they played in sort of helping you get to where you are, coupled, of course, with your drive, right, to actually do things and to be highly sort of entrepreneurial and creative. I think you've really left us with a very inspiring story. And I think it will inspire many, many individuals out there. So, Bavesh, once again, I just can't thank you enough for spending time with the Exchange. Our hope is that these discussions will really help folks understand not just what's happening with COVID-19, but also with other areas of science, but the process by which you intersect with science, the role that you play in moving science forward or in translating science in some way, and in a sort of a significant degree, how we're all engaged in this collective endeavor, right? And how important this collective endeavor of science really is for all of us. So thank you so much for spending time with us in the Exchange.
You're very welcome. It was a great pleasure.